So today I will show you the bare bones of how to analyze microarray data. So we are going to use these three libraries. The first one, tidyverse, in general to work with the data, but these are the two important ones. Mostly this one, limma, will do to fit the data, and then this oligo is mostly annotation. So we just go ahead and load those. The first thing that we want to do is to load the raw data, and that's what we have here. In addition to the raw data, we have this pdata or phenotype data that is just a very simple metadata table where we have the file names and the file that we are going to load, and I conveniently name them as the samples themselves, like control 1, 2, 3, 4. We have the different samples, control 1 to control 4, and then we categorize each one depending on what type of samples they are. So we have 4 controls and the 3 experimental. What we do is load that data. I'll run everything, and we use the information that is in this pdata file to look for the names of the files and then pass it to this function that is probably the most important one, that is doing the background correction, the normalization, and calculating the expression of the flow. So that gives us this eset object, and we can look at it as a table here. So we have this table where we have for each of the files the different probes as rows, and each value is the intensity of that probe. And here is all the files that we are analyzing. So I have here this AffyData script and put everything in the same folder. Here is our metadata pdata file, and these are our raw data cell files. So now the idea is to feed this to a model and then do some statistics. I will just run all of this. So the first thing that we have to do is to feed the linear expression model. So we are going to give that table that we are looking here with all the intensities to this function, and we are going to ask it to apply a linear model using the following design, and we create it with these lines here. And essentially we have the 4 first samples that will be control and the last 3 that will be experimental. Here we have 2 conditions, like the experimental and control, so there is only one contrast. So after fitting the model, what we want to do is look at the statistics, and then we do this calculation of the p-values for the difference between the observations in these 2 conditions. And after that, we can retrieve from that fitted model with the statistics all the table with all the values that is given here in this allstats table. So here we have for every probe the log-fold change between the conditions, the average expression, some statistics, and we have the p-value and the adjusted p-value or q-value for all of our probes. So this is pretty much everything. The last thing that I want to show you today is a little bit the annotations. Now we are going to load this annotation file. So for each probe you have to what gene is being matched. These files are provided by now Thermo Fisher. So you have these gene expression analysis files that we are using, I think. So we are using MoGene, one of these. So that's what I downloaded, and that's exactly this file here that you can get from there. So we are going to read that table. It's pretty cumbersome a little bit how they design it, but that's why I in this part here I'm isolating some variables. So this is how it looks like, the annotation. So for every probe set you have the name, and then you have the chromosome of the gene, the strand where it starts, where it ends, and then here a bunch of annotation, and including the gene symbol and other stuff. With this last line here I sort of extracted that data, so just to have the gene name, a little bit of the description, and the type of RNA that we are talking about, either protein coding, antisense, and so on and so forth. So now what we want to do is add this annotation to these 2 tables, to these statistics and this expression set, and this is what I want to show you last. So we will do something a little bit different. Instead of using all the probes, we are going to select the probes that encode for protein coding genes, and this expression eset table we are going to choose only the probes that match to protein coding transcripts, and we are going to do again the fitting and the statistics, and then we are going to add the annotation. And so we will end up with this allstat and this filter datasets that have now incorporated the annotation. So here is the allstats. So for example, this is the log4 change, average expression, and adjusted p-value. And then what we can do is look at the raw data or background corrected and normalized data for this transcript, for example. If we go to the other table that we generated, this filter, and we can look for that pro. Right, so that's it. Don't forget to watch the future videos where I give you some tools to look at the QC, the quality control of the raw data, and also check my next video on how to incorporate more than two variables into your design and how to make multiple contrasts in more complex analysis. With that, I hope this was clear.